Well, okay, so it is already 3 minutes past 2, so it's time to start our Sano Computational Medicine seminar. And today we continue seminars cycle about presentation of research of our PhD students at Sano. And today, as a speaker, we have Rosmary, Rosmary Blanco. Rosmary is working in Sano on neuroscience to understand the behavior of this type of phenomena, and also to explain how they are related with human behavior. Rosmary came to Sano with his Master of Science degree in medical and molecular biology from Italy, from University of Verona, and he, besides of this education, he also spent some time in clinical setting related to neurology, to neurosurgery, as well as rehabilitation. So, and research she is performing at Sano are related strictly to this area. You will hear about this in a moment. If you have a question, immediate questions, which is answer to which is really necessary to understand the rest of the talk, please ask this question on chat or on questions and answers, and I will try to stop Rosmary and ask this question. And of course, after Rosmary's talk, as usually, we will have time for discussion, for suggestions, as usually, which is important part of our meetings on Monday. So that's all as introduction. And Rosmary, please, screen is yours.
Thank you, Marian, and hello everyone. Let's start. Today I will discuss the advantage and the challenge of multimodal neuroimaging, specifically focusing on the integration of EEG and NIRS measurement. I will explore how we can translate the identification of typical brain patterns from medical devices in lab setting to wearable technologies in the real world. And I will present the results of my current research and outline my plan for completing this study. So in the last year, there has been an increasing interest in multimodal neuroimaging due to the recognition of the clinical benefit of multimodal data and the better access to hybrid devices. So simultaneously imaging measurement or the combination of separate measurement such as EEG and fMRI or EEG and NIRS has emerged as a prominent research area. Multimodal neuroimaging can overcome the limitation of single modalities or can reveal association among different imaging sources. However, different techniques depend on different source signals across various temporal and spatial scale, requiring sophisticated algorithms and computational methods to integrate diverse data. EEG is one of the most used neurotechnology for studying brain activity and diagnosing neurological disorders. It captures synchronized oscillatory brain patterns, providing a direct measure of neuronal activity. And the emerging perspective of neurodegenerative disease as oscillopathies make it a well-established technique for assessing brain functionality and its correlation with various pathological conditions. Moreover, collaboration across disciplines has led not only contributed to advancement in neurotechnologies, but also has led to the emergence of new fields in medicine, such as remote monitoring or telemedicine. So new applications in the medical field are emerging, including long-term at-home sleep monitoring or seizure monitoring or brain-computer interfaces for rehabilitative applications. And thus my research questions are how can we relate and effectively integrate the information from multiple estimates to obtain a comprehensive understanding of brain function, and how are the spatial and temporal dynamics of electrical and hemodynamic activity shaped by the underlying structure. And given the development of new neurotechnologies and their application in medicine, can we use clinical grade devices and the multimodal approach to identify EEG-based features that represent distinctive patterns of neurodegenerative pathologies? In my hypothesis, multimodal approach and effective data modeling can capture the complex spatiotemporal patterns of brain activity, crucial for advancing research in complex systems like the brain. And this method can enhance the detection of characteristic brain patterns indicative of neurodegenerative disease, potentially enable the development of innovative solution for diagnosis, monitoring, or guiding intervention. So in this context, network neuroscience offer a valuable approach to model and investigate brain dynamics, as well as to explore the potential of multimodal approach in inferring brain function. Thus, to address these questions, graph-based analysis was used to characterize the topological properties of electrical and hemodynamic brain network. The multi-layer network approach was employed to combine EEG and fMRS signals. Graph signal processing was used to investigate the relationship between the structural connectome and the electrical and hemodynamic functional data, and a multimodal approach integrating cognitive, behavioral, and EEG data was employed to enhance the performance of a machine learning model in distinguishing mild cognitive impairment from healthy control. But what are EEG and fMRS? They are two non-invasive neuroimaging techniques with EEG capturing directly neural electrical activity, while fMRS indirectly detecting the hemodynamic responses induced by neural activity. Combining these two techniques has become common in neuroscience because of their non-invasiveness, portability, and cost effectiveness. So EEG capture the macroscopic temporal dynamic of brain electrical activity. Its primary sources is the synchronization and summation of excitatory postsynaptic potential of the dendrite of the pyramidal neurons, and the current coming from a large population of neurons generate a voltage field that spread through the brain tissues due to volume conduction, and is detected by sensors placed on the scalp. fMRS is an optical spectroscopy technique that uses light injection and detection point to measure hemodynamic fluctuation in the cortex. Based on neurovascular coupling mechanisms, it measures change in the mood oxygenation in the region of activated cortical area. Thus, change in light intensity are measured as optical density and are related to concentration change in hemoglobin that are then converted in oxy and deoxyhemoglobin through the modified Beer-Lambert law. So to answer the first question, we investigated the correspondence between EEG and FNIRS network for resting states and task-related conditions, and assessed whether the multimodal approach of the integrated information from both modalities outperformed the unimodal approach. I used the synchronous EEG and FNIRS recordings collected during resting states and motor imagery tasks from healthy subjects. After a rigorous preprocessing pipeline to increase the data quality and eliminate the artifact, EEG and FNIRS signals were reconstructed in source space. And the source time series of both modalities were mapped on the same 3D space using an atlas-based approach, thus identifying 44 regions of interest. Then the network were constructed using graph analysis. Large-scale functional brain network can be mathematically modeled as a graph represented by a set of nodes and a set of connections between these nodes, whose pairwise couplings are summarized in the adjacency matrix, where the number of nodes corresponding to the brain regions extracted by the parcellation step, and whose arrangement define the network's topology. And here, the functional connectivity were calculated using Pearson's correlation for FNIRS and phase locked value for EEG, generating 7 connectivity matrices for each subject, 5 for EEG for the 5 typical bands of EEG, and 2 for FNIRS for the oxy and deoxyhemoglobin. And this procedure was repeated for the resting states and left and right motor imagery task, resulting in 21 functional connectivity matrices in total. One appealing aspect of graph model is that graph theory offers many objective data-driven measures to characterize the topology of the network. And here we choose the global topological measures of small world, global efficiency, global clustering coefficients, and characteristic path length, as these measures reflect the 2 key aspects of brain organization. First, that the integration relies on communication between specialized brain regions through a network of connection resulting in information flow. And second, that the segregation of specific brain regions and their connection are essential in integrated function. As measure of functional integration, the characteristic path length indicate how quickly information can be transferred across the network, while the global efficiency indicates how efficiently the information can be shared throughout the whole network using the shortest path between the nodes. And as a measure of functional segregation, the clustering coefficients reflect the tendency of nodes to form a local cluster with local interconnection, thus facilitating specialized information processing within the clusters. Thus, the small world network topology reflects this optimal balance between functional integration and segregation, which is crucial for cognitive functioning. And here was quantified through the small world propensity. we observed a small word propensity for both modality in the three conditions, supporting the assumption that the small wordness is a universal principle for the functional wiring of the human brain, regardless of the different imaging techniques and brain conditions. We also observed for EEG higher clustering coefficients than FMIRS networks, while both modality exhibited a similar path length value, indicated that the electrical networks have a better ability to form a subgroup of interconnected nodes in the network, but also that both EEG and FMIRS exhibit an efficient information transfer across the network, thus resulting in a small Notably, during the resting states, the alpha band exhibited higher clustering, aligning with its predominance during the period of rest. In contrast, during the task, we observed the increase in the clustering coefficients in the beta and gamma bands, which are frequency closely related with the motor imagery task. Also, our observation revealed higher global efficiency for EEG network compared to oxyhemoglobin network, and only during the resting states. And this suggests that EEG may be more sensitive to detecting subtle pattern associated with resting brain dynamics. On the contrary, during the task, no differences were pointed out, suggesting that the two modalities are more similar when increasing the synchronization of neuronal oscillation for sustained brain activation, like during the cognitive task. But to investigate whether the integration of information derived from different sources outperform this unimodal approach in characterizing the topology of the whole brain network, a multi-layer network analysis was conducted. A multi-layer network consists of several distinct classical networks, each one encoding a specific type of information about the system. So while a standard network can be represented by the adjacency matrix, to capture the complexity of the multi-layer network, higher dimensional structures are necessary. Thus, a multi-layer network comprises different networks encoded by layer, each represented by an adjacency matrix. And a common method for representing these networks involves flattening this rank 4 tensor into rank 2 tensor known as a supradjacency matrix without loss of information. The supradjacency matrix is represented by a square matrix where the diagonal block represents intralayer connection, while the off-diagonal block represents interlayer connection, the connection between the different layers. But when modeling the human brain, the primary approach are based on a special case of multi-layer network, which is the multiplex network. In this model, the same node is replicated on all layers, where it exhibits different connectivity pattern depending on the information encoded by each layer. This interconnected multiplex topology includes links across layer, although each node in one layer is only connected to its corresponding nodes in another layer. But when treating a multi-layer network based on different modalities, the range of the link weight can be very different. So to mitigate potential biases due to the differences in link density across layers, a common approach is to use a correction scheme for the individual layers separately. And we opted for a data-driven approach for thresholding the original EEG and FMIRS functional connectivity. Specifically, we generated binary networks in the shape of the minimum spanning tree. The minimum spanning tree is supposed to reduce the influence of the spurious connection within the brain by considering a strong connection that don't form cycles or loop to enhance the specificity of network topological characteristics. And we constructed this full integrated multi-layer network by using the EEG and the SNIRS minimum spanning trees. As a result, for each participant, this full integrated multiplex network consisted of 7 layers. 2 layers encoding for FNIRS, each hemoglobin, oxyhemoglobin, and deoxyhemoglobin, and 5 layers encoding for EEG information, specifically the 5 frequency bands. Each layer contains the same set of nodes and have the weight of the interlayer connection set to 1, so identical to the intralayer connection. We then apply the same procedure to build the multiplex networks exclusively for EEG by integrating the 5 frequency bands, and for FNIRS by integrated the 2 minimum spanning tree for oxy and deoxyhemoglobin. All these multiplex networks were built again for the 3 conditions: resting states and left and right motor imagery. And then we compared the full multilayer with EEG and FNIRS multilayer through graph analysis. And we choose as a topological matrix the global multilayer graph matrix of modularity, global efficiency, and path length as a measure of the effectiveness of the communication. Also, we have the superadjacency matrix. These measures are calculated as aggregated value over the entire multiplex network. So similar to single layer network, characteristic path length and global efficiency are measures of integration in the network. But in the case of the multi-layer, these measures consider all possible paths between nodes, including those that traverse the multiple layers. The modularity quantifies the division of a network into modules or community, where nodes within the same module are more densely connected to each other than the nodes in different modules. And it provides insight into the functional subgroup present in the network. So we found that significantly higher modularity and global efficiency value and significantly lower path length value when comparing the full multilayer to each EEG and FNIRS networks in both resting states and task conditions, indicating a more pronounced modular structure and superior overall information transfer in the full multilayer. These results suggest that the integrated approach capture organizational pattern that may be less apparent when using only unimodal EEG or FNIRS networks. Okay. If at the global level an integrated multilayer network demonstrated its superiority compared to unimodal network, we were interested in determining whether at the local level integrating these 2 sources of information we could provide redundant or complementary information into brain functioning during the motor imagery task. And to achieve this, we calculated the nodal metric of eigenvector centrality to identify key nodes in the network related with this specific task. And the eigenvector centrality measures a node importance in a network while considering also the influence of the other nodes, which in the case of a multi-layer is the centrality of the node. It is connected to across all layers. So for each subject, the eigenvector centrality value were computed in each node, aggregated across layer to obtain one value per node. Then we systematically computed the differences in each task condition relative to the resting states as a baseline, enabling to capture variation in brain activity during the task. Note that showed increased eigenvector centrality value during left and right motor imagery task compared to the resting states indicate that this node become more central or influential during this particular task. And this was repeated for the full multilayer, EEG multilayer, and FMIRS multilayer separately. Then we extracted the cortical regions linked to motor imagery task according to the literature. These regions encompass specialized area involved in motor planning and execution, but also other associative areas such as the prefrontal associative cortex that is involved in motor planning and decision making, or the parietal associative area. And we found that the full multilayer capture more nodes activated during left and right imagery task than the single modality, suggesting unique contribution from each technique. This finding could be advantage, for example, for improving the discrimination between neurodegenerative disease and healthy population due to their complementary nature. But we also noted that specific cortical regions were consistently activated across different modalities, suggesting that EEG and FMIRS to some extent can provide similar information that can be also beneficial as results, for example, for developing application for brain-computer interfaces, for enhancing the classification accuracy of such this model. To answer the second question, I was interested in investigating the structure-function relationship in electrical and hemodynamic network. The relationship between functional activity and the underlying structural connectome is usually evaluated using whole brain diffusion MRI-derived connectome and functional connectivity at the desired time scale. That can be electrical, hemodynamical, and it was reported that the structure and function relationship is modality dependent, suggesting that functional connection estimated from longer time window overlap significantly with the underlying structural connection. On the contrary, a structural functional mismatch may arise with shorter time windows due to the transient synchronization desynchronization. So this raises the possibility that the alignment between the relative slower functional activity captured by hemodynamic networks and the faster functional activity captured by the electrical network may systematically vary across the cortex. One method, one relative new tool for analyzing the structure-function relationship is the graph signal processing. Graph signal processing provides a method for analyzing and representing graph signals in a spectral domain, where the graph is represented by the structural connectome, and the signal are, in this case, the source time series extracted from the synchronous EEG and SMIRS recording, having the same parcellation scheme of the structural connectivity. Therefore, the graph signal is essentially a vector in the space defined by node in the graph. Despite our data varying over the time, graph signal analysis focuses not on the temporal dimension, but on analyzing one signal at a time. In other words, each time point is treated as a snapshot or specific pattern rather than a continuous time courses. But to understand how brain function uses its underlying structure, we use the concept of graph spectrum and graph spectral embedding. The graph spectrum is determined by the Hagen decomposition of specific graph operators, such as the adjacency matrix or the Laplacian matrix, as in our case. Graph spectral embedding performed through Laplacian Hagen decomposition allow us to represent how nodes are organized based on their connectivity strength in lower dimensional space spanned by its eigenvector. And this approach optimizes the representation by minimizing the total variation of the signal across the graph. In this context, the total variation refers to how much the signal values vary across the graph, considering both differences in signal value at different nodes and their structural connectivity represented by the value of the adjacency matrix. So the eigenvector of the Laplacian represents different modes of variation within the graph that are called the structural harmonics, and the eigenvalue can be interpreted as the spatial frequency of the corresponding structural harmonics. And if we plot this eigenvalue, we can see that the first eigenvalue is always 0, which is a constant pattern across the brain, spanning across the whole brain. It is like a DC signal in classical signal processing. And the lower eigenvector exhibits slow variation, reflecting global structural connectivity and capturing the main connectivity gradient, such as connection in left and right hemisphere or anterior and posterior. Higher frequency instead are more localized patterns, often with a group of nodes rather than the entire brain, and exhibit rapid variation, showing less structural information. In this work, the graph is a group consensus structural connectome constructed from diffusion MRI. We computed the eigen decomposition of the normalized Laplacian of this structural connectivity and found eigenvector representing our structural harmonics. We then projected the functional signal, the EEG and SMIRS source signal into this space spanned by the structural harmonics by decomposing the functional signal through the graph Fourier transform. This transform decomposes the functional signal into coefficients indicating the contribution of each structural harmonic. Then the filtering process in the graph domain is applied to extract relevant frequency component by using the ideal low pass and high pass filtering, and choosing as a cutoff frequency the value that divide the power spectrum into two portions of equal energy. These filtered graph signal are then transforming back into the temporal domain by applying the inverse graph Fourier transform, resulting in a lower frequency component which are more coupled with the underlying structure, and a high frequency component which are more decoupled with the structure. The ratio between these two components yield the structural decoupling index, which quantify the level of decoupling between the structural and functional connectivity for each node. As a result, we obtain an SDI map for each subject for both conditions and for each modality. And the distribution of the SDI value across subjects were compared statistically. So our initial hypothesis aimed to determine significant differences in the overall distribution of the SDI value within modality for both resting states and task conditions. So we observed an overall differences in structure function coupling between EEG and FNIRS only for resting states, while no disparity was shown for the task conditions, implying that the modality exhibit more similar pattern of structure function coupling and decoupling when the brain is engaged in a cognitive task. When investigated the structure function relationship change within each modality for EEG, we showed that for resting states, higher coupling with the structure was shown for the alpha band, while for the task condition in the beta band, reflecting task-specific modulation of neuronal interaction. For FNIRS, during both conditions, oxyhemoglobin exhibited higher decoupling than deoxyhemoglobin, suggesting that this relationship is in any case closely linked with the demand for oxygenation. And when investigated the interaction between the two neuroimaging modality, we showed that oxyhemoglobin network was more aligned with the alpha band in the resting state, since no differences were pointed out between the two modalities. Contrarily, for the oxyhemoglobin, the relationship with the alpha band was of more decoupling. And during the task, oxyhemoglobin exhibited alignment with the delta and to less extent with the beta band, conversely to the oxyhemoglobin. Our second hypothesis aimed to determine how regional heterogeneity influence the structure-function relationship between modality for both conditions. And we observed a similar spatial distribution of the SDI value for both neuroimaging modality and condition, characterized by higher coupling in the sensory cortex, more evident in sensory motor and visual cortex, highlighted as the blue dot, and increasing the coupling in the association cortex, covering more frontal area and the posterior parietal associative area. And when comparing EEG and FNIRS at local level, this convergent pattern within the same array, within the same regions, varied in intensity, indicating a predominance coupling for oxyhemoglobin over the EEG band. But discrepancies between resting states and task condition were highlighted with fewer regions that were significantly different between the two conditions during the task. Moreover, differences in key regions specifically related to motor imagery task can be observed in the premotor cortex, parietal region, or dorsolateral prefrontal cortex, for example, is specific for beta and delta and gamma band that are related with the imagery, motor imagery task. And to further explore this distinctive pattern between modalities and relate this pattern to brain functioning, we categorized the brain regions based on their association with macro-scale intrinsic functional network, observing that a higher structure function coupling in the visual network and somato-motor network, and in contrast, a higher decoupling in the default mode network and attentional network is visible for both modality and all frequency band. In contrast, the frontoparietal network exhibited a divergent pattern characterized by higher coupling in lower frequency band and higher decoupling for the faster frequency band in resting states with an inverted pattern during the task, while for oxy and deoxyhemoglobin, this pattern is showed a similar profile. But since previous research has shown that synchronized oscillation across multiple frequency bands can best explain the well-established fMRI functional network, we incorporated a cross-band representation of the electrical network to gain further insight into the convergence divergence of EEG and FNIRS structure-function relationship. And this analysis revealed a lower correspondence between electrical and hemodynamic network in transmodal cortex, regardless of brain states, while specificity was pointed out for the somatomotor network during the task. So my last research question. This research aimed to address the urgent need for accessible and cost-effective diagnostic tools for early dementia diagnosis. Alzheimer's disease is a neurodegenerative disease associated with progressive cognitive decline, and it is the predominant cause of dementia. Early diagnosis of dementia or Alzheimer's disease is crucial for effective treatment planning to slow the disease progression. However, up to 50% of those living with Alzheimer's disease may not have received a formal diagnosis. And traditional diagnostic methods often face limitation. Neuropsychological tests lack sensitivity, have higher variability, or medical imaging are costly or are too invasive. So EEG biomarkers offer a cost-effective and objective measure of neuronal activity, even if most of the study on EEG-based biomarker for early Alzheimer's disease diagnosis rely on resting state experimental protocol, event-related potential have shown promise as EEG-based biomarker for dementia assessment. Event-related potential are patterns of brain activity time-locked with the specific stimuli or events. And this waveform pattern reflect the synchronized neuronal activity of brain response in response to the stimulus, and are characterized by their amplitude and latency at specific point on the frame, on the waveform. ERP are composed of different components that represent distinct stage of cognitive processing. Particularly, the P3 component is associated with attention and cognitive processing related to the stimulus evaluation. And this was shown that this component is sensitive to cognitive decline in Alzheimer's disease that can discriminate between mild cognitive impairment and healthy control, as well as Alzheimer's disease. It is not influenced by cultural background or educational level, thus represent an ideal candidate to develop a new tool for cognitive impairment assessment. Moreover, wearable EEG devices present a significant advantage for this purpose. They are portable, cost-effective, and user-friendly. Multimodal monitoring has proved to enhance the diagnostic capability in neurodegenerative disease, and AI-based technologies can significantly enhance the clinical decision support system through computer-aided tools. Here I will present the developed framework and the preliminary result of this study, as this is still a work in progress. And the framework is this EEG-based passive P3 application framework that aim to identify EEG-based features that differentiate between MCI and elderly individual. The framework is composed of different modules synchronized through digital triggers. The module synchronizer control the time synchronization of the EEG data and the event of the stimulation through the LSL bridge, as well as the real-time signal visualization. Then there is the module sensors that control the Bluetooth connection, streaming the data, the control the recordings, save the data in CSV file, and the module markers for the control of the image presentation and the triggers associated with specific type of stimuli. There is also an intuitive graphical user interface that allows users to easily add a new subject, initiate or conclude EEG recording or stimulation, and reviewing the data faster after the recording. So by integrating different data sources and employing a supervised approach, the aim is to enhance the interpretability and clinical relevance of the framework. Thus, multimodal data are included in the analysis by integrating cognitive motor function measurement with the neurophysiological metric of P3 component and EEG oscillatory patterns. The stimulation protocol is a non-invasive, passive 5-minute stimulation test during which subjects are required to view images containing positive, negative, and neutral valences. This kind of stimulation aim to assess how emotional stimuli of different valences impact the modulation of visual attention. The emotive stimulation protocol offers insights into the underlying neurophysiological processes associated with dementia by exploring the limbic circuit, including those involving amygdala and the hippocampus, which are affected early in dementia. The schematic of an ideal automated EEG-based system for early diagnosis of dementia has been proposed in recent years, very recently reviewed by emphasizing the state-of-the-art wearable sensors for neurodegenerative disease diagnosis and assessment. So, following this recommendation, the general workflow of this study is graphically depicted in this diagram. And from raw EEG data, the preprocessing step aimed to remove artifact, retain the relevant EEG frequency band by filtering. The feature extraction step was employed to extract domain-specific features with expert knowledge incorporation. I opted for a supervised approach, extracted the features with demonstrated promises, a potential biomarker associated with dementia, and specifically behavioral feature from the motor test and temporal domain and frequency domain features from EEG. Then feature selection using mutual information was performed, and a leave went out cross-validation model evaluated with the metric of accuracy, precision, recall F1, and confusion metrics was used for the model performance. So, as I said before, this is a very preliminary. These are very preliminary results. The data set comprise 18 subjects aged 65, 90, of which 6 healthy control and 12 MCI, classified according to mini-mental state examination and the Montreal Cognitive Assessment Test. For each subject, epochs of 1 second length were constructed from the raw data. Thus, a data matrix of dimension channels per time point per trials was constructed. After that, averaging across the trial dimension provide a single event related potential for each subject. The ERP component were automatically identified, and the features in time and frequency domain were extracted automatically. I tested 4 classifiers, those commonly used with EEG data and that achieved high classification accuracy in previous study. And here, the linear SVM outperformed the other classifiers, achieving the highest accuracy score. The model, the coefficients of the model provide also insight into how each feature influences the classification toward one classes or the other. And according to the literature, these features underscored the lower alpha and relative absolute power for MCI sample increased the P3 latency and a reduction on motor performance for the MCI subject. Finally, when predicting an unseen case, the trained model demonstrated a remarkable success in accurately classifying the new subject. So, to conclude, multimodal neuroimaging integrating EEG and SNIRS showed a significant potential in capturing organizational patterns that can be not apparent when using the individual modality, and highlighted its superiority in identifying the brain regions related to the specific task. Structure-function relationship showed the variation across oscillation and between conditions alongside the associated hemodynamic response. There is a regional heterogeneity in structure-function relationship, but this is systematic across modalities and conditions. However, discrepancies between the two modalities were shown for specific cortical regions depending on brain conditions and frequency band. The P3 framework identify features that align with those suggested as a potential biomarker for dementia and Alzheimer's disease, and the stimulation protocol proved effective in the generation of its related ERP component. The model's ability to accurately predict new cases demonstrate its generalization capability. So this is a work in progress. So I'm still working on improving the performance of the classification of the model by exploring other algorithms and hyperparameter tuning. The data collection is ongoing, and I am improving the pipeline by testing with permutation test the significance of classification score. I am adding further step in the analysis, such as statistical analysis and interpretability. When in the future, I'm planning to apply this pipeline to a larger data set of healthy elderly individuals collected with medical EEG equipment to track the physiological cognitive decline over time and compare it with that feature with MCI subject. And I don't know, this is an idea, employing a transfer learning approach to enhance wearable EEG data classification performance for dementia detection by using noisy raw EEG data collected in the real world in daily life. Here is a list of publications I have produced since joining Sano, which have results from collaboration within and between teams. And to foster further discussion, I would like to pose few questions to the panel. But maybe I can be back in this slide after my very thank you. I would like to thanks everyone who helped me make this project possible. Natalia for assisting me with the ethic committee's request, Kasia and Emma for helping me with finding senior home, Carolina, Taras, and Monica for their invaluable assistance in communicating with seniors in Polish, but also much more, and especially Maria, the manager of the senior home, and all the residents, of course, who volunteered to participate in this study, and all people in Sano who were involved. So very thank you for your attention. I'm sorry I was too long, but I'm happy to answer any question that you may have.
Rosmary, many thanks for your talk, which is full of also technical details. And now we have time for questions. But what I would suggest that it would be great if you could move well, because for the first time this talk of our PhD students in Sano, we had the situation when, besides of, let's say, a kind of report from these research activities, there are also some questions which probably answering them is very, let's say, important for further research. So let's this slide will be kept well as long as it will be possible. Maybe some questions will let's say, will force to change to another slide. But anyhow, now I open discussion. So please go on with your questions, suggestions, and maybe answers to questions which were posed by Rosmary.
I am mainly interested in the second and the third question. If anyone could help me, because I'm not a data scientist, so I'm a project. I'm studying a lot, okay, but your suggestions are valuable, of course.
Yes, I understand that you had also one of co-author of your paper is Szymon Mazurek, and well, he somehow is expert in this. Do you think you will continue collaboration with him?
Yes, of course, and I hope. I have collaborated with Shimon with several paper, and we can sure continue working together.
I have a question about your investigations, about your research, which was related with these old people, and how it was organized, and how you have managed to collect this data.
From a technical point of view, you mean?
Yes.
Okay. It was very challenging at the beginning because these devices don't provide, if not their own tools for streaming the data from these devices. But at the end, I was lucky and I found a third-party software that could stream the data, the raw data from these devices. So I integrated this software in my framework, and it was a collage of tools, and it open source. All tools I used are open source, and I would like to highlight the fact that the tools doesn't work only with this kind of device, that is the Muse EEG devices, but can work also with other devices. For example, we have at the office the OpenBCI. I didn't test it and check it with this other device, but it should be possible.
Thank you. Now we have in queue Wiesław. Well, before I will invite Wiesław to ask the question or comment, because we have I see on the list of participants the new persons. So Wiesław is Professor Wiesław Nowinski, who was working for quite long time in Singapore, and who is author known as, among others, as the author of Atlases of Brian. So just information for newcomers. And this floor is yours.
Thank you, Marianne. Thank you for your kind introduction. Okay, Rosmary, it's very nice work, lots of results. Regarding the first question, so my question is, of course, quality depends on the input data. So what sort of device do you use for measurements? How many sensors or electrodes in your input device?
The device has only 4 channels, but I integrated some channels, and at the end, I did a different combination of these 4 channels, and at the end, I get 10 channels. So it was another kind of generative signals than using a gun.
Because on some
I pulled simply pulling the channel together with different combination.
Because on some slides you presented, it looks at there are around 40 measurements, but of course there are some devices with so-called like here, dense EEG with 256 electrodes, which of course give you a better sampling, and definitely you get better results. So I believe that having a better quality of data will give you a better results and maybe easier way to process them.
But honestly, I don't have bad results. I worked a lot with the signal processing of the raw EEG signals as captured by these devices. But this is the result of, this is my result of ERP with only 100 average stimulation, and it is very good. The waveform of the ERP measured from typical traditional medical devices are exactly the same. So I was surprised too, but it was the first thing I was sure before continuing with the rest.
Yeah, regarding the second question, of course, some other modalities to assess dementia, AD, MCI, like magnetic resonance, and even earlier, some signs might be detected using PET imaging. How do you think, maybe it's too early, how EEG compares against these two? Is it possible to using EEG and oscillopathy to detect dementia earlier than this state-of-the-art technologies? Have you tried to compare? Or do you
No.
plan to compare?
Why not? I am here also for asking how to proceed or continue my research from that point, because till now I was working on this EEG and FNIRS comparison, also for a better or a reasonable choosing of EEG instead of FNIRS devices. And then I'm very starting now with this project, so I have plenty of possibility to compare everything we want or to do another research. If you suggest something, you're welcome.
It looks to me that you have a very nice platform, and also on the other hand, there is a big market because, of course, wearable or portable EEG is available commercially for home market. So I believe people can buy and measure their own EEG. Like currently using smartwatches, we can measure blood pressure, pulse, and so the same can be done with EEG and with early detection of dementia. So I think that if you could compare with your results with MR and maybe with PET, It might open a great commercial opportunities for home market with portable wearable EEG. Does it make sense? ECG.
Yes, it is not the actual aim of the tools, because I was thinking at the tools that can be used by clinicians and not by the patients itself or their caregiver. The idea was to provide the clinicians one different tools that can help making them decision about cognitive assessment behind the neuropsychological test, for example. So advice that can be included in the early steps of when the one subject should be screened if he needs or not a second step screening like neuroimaging, MRI, FMRI, or PET. Something that can be useful and that can provide.
Well, don't worry. I hope that Mary will come back in a.
Sorry.
Yes, you are
I
back.
don't know if it's my connection problem. So what I'm saying is that the idea when I developed these tools is to give one tool useful for clinicians to make objective decision about early screening of cognitive assessment, so they can decide if this subject needs to be investigated further with PET or magnetic resonance, instead of using only neuropsychological test. At the end, in 5 minutes, you can do all the process, all the simulation. This is the advantage too.
Yeah, of course, I don't want to ruin your thinking, but as Marian mentioned, I work for a quarter of century in Singapore, and over there research is very pragmatic. So meaning what we try to do, we try to be useful. And let's say if I would be in your shoes, my thinking would be, okay, if I like to do a tool for clinicians, I need to go through a regulatory process. And 1 or 2 weeks ago, we had a nice talk how difficult it is to go through FDA. If I go to home market, the market is bigger, and you don't need to go through regulatory process. And your contribution to the society and to Sano budget would be much, much higher. So this would be my thinking.
Okay.
Well, it
Okay.
is very good thinking from.
Very exactly.
Okay, another comments. You have very nice title, emerging technologies. So for me, emerging technology is wearable MEG. Have you tried to think about applying these technologies, or some of your methods may be also applicable for MEG, magnetoencephalography?
Yes, I don't think so. MEG is very complicated instrument. I don't know if it could be portable or
It is
wearable.
portable, but what I'm saying, portable EEG is not, I would say, emerging, but MEG is emerging. And of course, I believe many problems would be the same. You have the reverse problem to reconstruct the dipole and so on. So therefore, I'm just thinking that you can think about the potential extension. And another also questions or comments about extensions. You are talking about application for dementia. What about application for brain-computer interface, which also will have a great and wide market?
Yes, I can start from here. I'm very interested, of course, as a biotechnologist in BCI application, and I'm trying to develop a tool for neurorehabilitation, but I haven't a lot of time at the moment, so I'm having fun sometimes, but but yes, something quite ready with this EEG band and application written in Unity, gamified application that can be used for rehabilitation. Yes, I am thinking about this.
It looks to me that you need a team of people because there is too much work for you alone.
Thank you.
Okay,
And
so good
I
luck.
don't remember the first question, sorry.
First question meaning?
Oh no, I just answered. Yes, sorry.
Yes. Well,
Thank you, Liz.
I see that we have with us Alex. Alex, would you like to comment research of Rosmary? Or
Comment for what?
yes, to add something because well,
It's difficult. We have been talking for a while. I don't know. Maybe the story of comparing the P300 with will be good to look what the Brain Trip. There is this Slovenian Malta startup that they were doing it for more detailed EEG. I don't know if there is some data set available and see if the P300 from them test is.
No, I look for other data set for comparing our results, but no.
Because I know that they are doing exactly this, but I don't know if it's available.
No, I don't think so.
Well, maybe it's worth maybe to contact them and ask them if they're willing to give something.
Well, so thank you very much. And now, Rosmary, could you please display this slide with list of your publications? I have a question about it because there was a list of your one of the last slides. Oh, yes, here. So you have a number of submitted papers, as I understand. This in Journal of Computational Science. It is in review.
Yeah.
Yes.
Yes, it is.
And it is the result of being selected during a previous ICCS conference, or you have just.
No, I've extended the previous paper published in ICC conference, and it is those of multi-layer. So for the paper of ICCS, I simply compared the through graph analysis, EEG, and definite network. Then I included adding all the multi-layer network analysis, and this is the paper that is under review in computational science.
Yes, clear. So, and well, to which extent the journals which you have published up to now are typical for this type of research? Because it is is probably the first one, which was about biomedical imaging is probably the best for this type of research, yes? Or
I don't know. I was invited to publish in Journal of Computational Science by extending my previous paper, but they enjoyed the paper. The editor and the reviewers also said very nice things about the paper, and they required only minor revision to be published.
Mhm.
So I
Well,
think
well,
yes, because I didn't show during this seminars all the process, all the computational method that are included with more detail in the paper.
yes, I understand that there up to now, at least there are at least two outcomes of your research. On the one hand, you have something like you have a method which you have already elaborated, and you can apply this method to different situations, to different illness. This is on one hand. On the other hand, I understand that what you have done with these elderly people, it is something. It's more than method. It is just some research. And well, are you going to communicate to these people the result of your research?
No.
No, why?
no, sorry, but I'm not sure to have understand your question. Could you repeat, please?
Yes, because well, these people gave you some, let's call this data. Probably it would be good to let them know what usage of this data was done. So I think that you should stay in contact with
you mean the resident in Doma Signora?
Yes.
Yeah, no, but I'm still collecting data, so we are friends. They wanted to know sometimes how is my brain? Is it still working or no? I have dementia or not? But I cannot say nothing.
I can imagine that they are really interested as everybody. Wisław is back with his hand up.
Yes, yeah, because
Thank you.
it's resulted from your comments about the current results. It looks to me that again, it's based on my Singapore experience, looking at the changes in EEG signals, and you have a method for analyzing them. Is it a space to file a patent? Because maybe you are proposing a novel way of doing something which so far no one has done, and it might be cheaper than the current existing methods. Again, it's not a question to you, but maybe question to Marian and to others.
Mm-hmm. Well,
for me, it would be wonderful if we can patent the tools. Why not? But
First,
I'm not an expert, so I need for sure someone.
No, you are an expert because you have to check whether in the literature no one has done the way you did it. Okay? So you might say.
Sorry.
Yeah.
Yes. So I think that it is this was probably I hope that we together with Rosmary will have a chance to talk at Sano how to make reasonable usage, scientific and not only scientific. So it's so I'm very grateful for your questions and suggestions here.
No problem.
Well, we have reached almost time devoted. We have not almost. We have really reached the time devoted to the seminar. So, Rosmary, many thanks for your talk. Many thanks to Wiesław and to Alex, and thank you very much. And I would like to invite all of you to next seminar of this series. It will be in 2 weeks from now, on the 1st July, because, next Monday, we will have at Sano Children's Day. So, well, I think that there is still opportunity to subscribe to register for this Children Day. So, well, you are invited to our seminar in 2 weeks from now. Many thanks.
Thank you. Thank you. Bye-bye.
Thank you.
Bye.